Now, muscle physiology, important questions. What is the structural and functional unit of nervous system? It's neuron. What are neurotrophins? Give example. Neurotrophins are number of proteins that promotes the growth of neurons responsible for maintaining the functional and structural integrity of axon. Example, nerve growth factor, brain derived from neurotrophic factor, neurotrophins 3, 4 and 5, insulin like growth factor 1, etc. What is functional organization of neuron? Now cell body along with the dendrites is the receptor zone responsible for the graded electrogenesis, non-prolongated local potentials are integrated here. Axon, initial segment, generation of the propagated action potential transmits propagated impulses away from the cell body to the nerve endings, all are non-transmission. Synaptic knobs, nerve endings where action potential causes releases of synaptic transmitters. Which part of neuron has lowest threshold for excitation and why? Initial segment of the axon because low voltage gated sodium channels are highly concentrated in this region. Difference between the myelinated and unmyelinated neurons. Myelinated neurons and unmyelinated neurons. Myelinated neurons, one cell membrane is coiled many times round in the axon forming myelin. Axons are simply buried in the Schwann cells without wrapping the myelin. Examples are preganglionic fibers of autonomic nervous system. Example, all postganglionic fibers of ANS. Nerve fibers of somatic nervous system are more than 1 micrometer in diameter, whereas nerve fibers of somatic nervous system less than 1 micrometer in diameter. Conduction of nerve impulse is faster in myelinated, that is 550 to 100 times than in the unmyelinated fibers of same diameter because of saltatory conduction. Con- Conduction of nerve impulse is slower because of the continuous process. What is myelin? Two lipid layers sandwiched between the third layer of protein. In myelinated nerves, why action potentials are generated only at the nodes of Ranvier? Because no voltage gated sodium channels are highly concentrated in this region. What is multiple sclerosis? There is a patchy destruction of myelin in the CNS. It is crippling disease associated with a delayed or blocked conduction in the demyelinated axons. What is glial cell? What is the glial cells and what are its type? Which cells are responsible for forming the myelin around the axon within the CNS? Cells which supports the nerve cells are called glial cells. Types include microglial cells, which phagocytic cells remove the damaged cells, and foreign invaders come from the bone marrow and enters the CNS from the meninges. Astrocytes, most numerous, found throughout the brain, joining the blood vessels. Function, it supports transport. Transport mechanism. Inflammatory and reparative infections helps in forming the blood-brain barrier. Optimal concentration of ions. Neurotransmitters in homeostasis in the brain neurons, as plays an important role in learning and memory by forming the neuronal synapses. Oligodendrocytes forms myelin around the axons within the CNS. Note, astrocytes and oligodendrocytes together are called macroglial cells. List physiological properties of the nerve fiber. Excitability, it follows all or none law, refractory period, accommodation, and conductivity. Name factors which affect the excitability of the neuron. Strength and duration of the stimulus. Extracellular calcium, if decreases, increases membrane permeability to sodium and increases excitability. What is the unit to which all or none law is applicable? It applies to either a single nerve fiber or an individual muscle or the heart. To the heart, the unit begins entire arteria or entire ventricle. Give physiological significance of refractive period. This puts an limit to the frequency at which the nerve can generate or conduct impulses, the maximum of which is not more than 1000 per seconds. 1 by refractive period, that is equals to 1 by 1 milliseconds. In saltatory, what is saltatory condition? Saltatory condition means leaping or jumping. It is a feature of myelinated nerves and can conduct impulse 50 to 100 times than an unmyelinated nerve fiber of the same diameter. Define orthodromic or antidromic conduction. Impulses normally passes along the axon to their termination. Orthodenic conduction or conduction in the opposite direction is called antidromic conduction, seen in the sensory nerves supplying the blood vessels. Name factors that affect the spread of conduction in a nerve. It is directly related to the amount of myelination or axon diameter or to the temperature. What are the types of nerve fibers and give their functions? Nerve fibers, myelinated motor or sensory of spinal nerves, alpha, A beta, A gamma, A delta, B or C. A alpha function is proprioception and somatic motor. A beta touch pressure and motor function. A gamma motor to the muscle spindles. A delta pain, temperature and crude touch. B preganglionic or autonomic nerve fibers, whereas C are unmyelinated in dorsal root, pain, touch, temperature and conduct impulses generated by the cutaneous receptor, whereas sympathetic postganglionic sympathetic nerve fibers. Give clinical features of nerve fibers based on the sensitivity to hypoxia, pressure and anesthetic agents. Sensitive to hypoxic, most susceptible with beta V fibers. Clinical feature alteration in BP, heart rate and respiration. Sensitivity to pressure, A fibers, temporary paralysis, Saturday night palsy. Sensitivity to local anesthesia, C fibers used for stretching cut wounds. What is the compound action potential? Give its feature. It is an extracellular or biphasic recording of an action potential from a mixed nerve made up of the families of nerve fibers with appearance of multiple peaks. In general, group A fibers make maximum contribution to it and group C are the least. Factors affecting type of the fibers in a particular nerve. Note, none of the peripheral nerves contains all fiber types. Strength of the stimulus determine the shape of the compound action potential. Distance between the stimulating and the recording electrodes. If distance increases, greater separation between the peaks. What is Wallerian degeneration? The degenerative or destructive changes in the distal part of the cut nerve fibers is called Wallerian degeneration. What degenerative changes occurs in the neuron following the complete transection, that is fifth degree injury? Changes in the nerve cell body, retrograde changes begins within the 40 hours and reaches maximum within 15 to 20 days. Chromatolysis, Neissel's granules break up into fine dust. Cell organelles fragmented and disappear, nucleus displaced to the periphery. Changes in the distal stump, Wallerian degeneration. The process starts within 24 hours. Changes in the action potential and ability of the nerve to conduct the impulse decreases markedly. Axis, cylinder and myelin sheet break. Macrophages start digesting the debris. Nucleus of the Schwann cells start multiplying and fill up the endothelial tubes. This process takes 3 months. Change at the site of the injury. Schwann cells grows at 1 to 2 mm per day and can bridge the gap up to 3 cm. What degenerative changes occurs in the neuron after transection? Recovery of the nerve cell body begins 20 days and it is completed in 80 days. Changes of the distal stump and at the site of injury. Axis cylinder from the proximal stump elongates and then grows out in all directions. Initial rate of growth 0.25 millimeter per day, but once it enters the distal stump, it becomes 3 to 4 millimeter per day. It's about 15 days. Schwann starts laying down the myelin sheet around the successful fibril, which is completed within 1 year. Related viva voice questions. Define retrograde changes, that is chromatolysis, following injury to a nerve. When do retrograde degenerative changes begins? After transection of a nerve, when do the regenerative changes begins? Initial rate of regenerative growth following an injury to a nerve. Schwann cells can bridge the gap up to what extent and after an injury of nerve. What is neuromuscular junction? Junction between the motor nerve fiber and skeletal muscle fiber is called neuromuscular junction. What is motor end plate? It is a specialized part of muscle sarcoplasm. Each vesicles of synaptic terminal of neuromuscular junction contain how many molecules of acetylcholine? It's approximately 10 power 4 molecules of acetylcholine. What is the width of synaptic cleft in neuromuscular junction? It's a 5 to 50 to 100 nanometer. It is filled with the extracellular fluid. Name the receptors found at the postsynaptic membrane of neuromuscular junction. It is a nicotinic acetylcholine receptors. Give sequences of events in neuromuscular junction, nerve impulse transition. Nerve impulse or action potential reaches presynaptic nerve ending, increases permeability of the presynaptic membrane to calcium, decreases flow of axoplasm, fusion of synaptic vessels with a terminal membrane, synchronized release of 10 part 6 molecules of acetylcholine into the synaptic cleft. Acetylcholine attaches to nicotinic acetylcholine receptors on the motor end plate, increases permeability of the motor end plate to sodium mainly, and depolarization of the postsynaptic membrane, generation of an local action potential or end plate potential at threshold value, that is 30 to 40 millivolt of end plate potential, generation of action potential, that is spike potential, which is 120 to 130 millivolt, propagated muscle action potential, and muscle contraction will take place. What is miniature end plate potential or MEPP and how it is produced? It is EPP, end plate potential at rest. Normally it's up to 4.5 millivolt and it is produced due to the Brownian movement of the axoplasm. Fusion of some vesicles with a nerve membrane. Release of acetylcholine is in very small amounts, less than 10 per 6 molecules. What will happen with inhibition of membrane end plate potential? There will be production of gained end plate potential GEPP up to 10 to 12 millivolt due to the release of more quantity of acetylcholine. It is responsible for fibrillation of muscle. Give clinical significance of neuromuscular junction. Why blockage of neuromuscular junction is required? Blocking neuromuscular junction muscle relaxation, which helps in surgical operations by providing surgical operation by providing open field, reducing the movement of electric shocks or during psychotic patients, and reduction of fractures. How a blocking of neuromuscular junction can be achieved? Botulinum toxin interferes with the synthesis and release of acetylcholine. Drugs which antagonize the action of acetylcholine on motor end plate by competitive inhibition. Tuberculin, which is an antagonist to curare or anticholinesterase neostigmin by persistent depolarization. Suxametronium. What is myasthenia gravis and how it is produced? Name commonly affected muscles. It's an autoimmune disease, causes formation of circulating antibodies to nicotinic receptors. Nicotinic acetylcholine receptors feature rapid onset of fatigue, markedly generalized weakness of muscles. Most commonly used muscles are extraocular muscles, facial muscles, so swallowing and mastification muscles. In severe form, patient may die from paralysis of respiratory muscles. Name muscle protein and give their functions. Contractile protein. Actin and myosin generate force during contraction. Relaxing protein tropomyosin and troponin relaxes muscle. Structural protein titin, alpha actin, myomyosin, nebulin, and dystrophin contribute for elegant stability and elasticity and extensibility of the myofibril. What is the contractile unit of the skeletal muscle? Substances including between the two adjacent Z lines are called sarcomere, 2.5 micrometer in length at rest. What is the component of sarcomere? Thick filament made up of myosin, formation of A band, whereas thin filament made up of actin, troponin, and tropomyosin, formation of I band. What sarcotubular system is made up of and what are its function? T system or transverse tubular system, inward directed extensions of muscle membrane into the muscle fibers at A and I junction. Function rapid transmission of action potential from cell membrane to other fibrils in the muscle via dihydropyridine receptor. Longitudinal sarcoplasmic reticulum with a dilated terminal cystae rich in glycogen and calcium. Functions concerned with calcium movement and muscle metabolism via Raynaud receptors. What is triad? Transverse tubule with the two terminal cisternae. It is found at the AI junction. There are two triads for sarcomere in skeletal muscle. What is excitation, contraction, coupling? It is a process in which depolarization, excitation, and electric phenomenon of the muscle fiber initiates contraction, that mechanical phenomenon. These two phenomena coupled or joined together by the coupling agent calcium. What is the role of calcium in muscle contraction? Calcium binds to troponin. Tropomyosin moves laterally, exposes the binding site of the myosin head on the actin. and activates enzyme ATPase on the myosin 8, catalyzing the breaking down of ATP energy production, activates sliding filament system and contraction of actomyosin complex. For more questions, follow my channel. If you find this video helpful, please do like, share, and subscribe to my channel. If you have any suggestions, let me know in the comment section. Thank you.